Good afternoon. I am Dr. Kara Wilder, a technical writer at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Neural Progenitor Cells, Better Biological Models of Neurodegenerative Disease, presented by Dr. Brian Shapiro. Dr. Shapiro is a technical writer at ATCC. In today's webinar, he will provide an overview of neural differentiation. He will then explore how ATCC-NPCs can be differentiated into these lineages and used in toxicological studies, focusing on the performance of the Parkinson's disease-derived NPCs. If you have any questions for Dr. Shapiro, please use the chat function available through the webinar program. Questions will be answered at the end of the presentation. Any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website at www.atcc.org. With that, I would like to welcome Dr. Shapiro.
Thank you, Kara. Good afternoon, everyone, and thank you for joining us for this webinar. The last time I presented, I revealed that ATCC was taking on new efforts to validate its neural progenitor cells as tools for toxicological screening. Today, I'll present ATCC's Parkinson's disease-derived neuroprogenitor cells. Like other neuroprogenitor cells, these can be differentiated into multiple neural lineages or used in their undifferentiated state to investigate the pathogenesis of Parkinson's disease or used for neurotoxicity studies. But before I talk about these innovative neuroscience tools, I'd like to say a few words about ATCC. Scientific advancements are critical for unlocking incredible breakthroughs in global health. ATCC has provided credible biomaterials to the scientific community for over 90 years to help achieve these breakthroughs. We're continuing to cultivate that collaboration among scientists and across disciplines by sharing the credible information that is essential for accelerating innovative research that leads to incredible high-impact results. To this end, we pledge to bring scientists together to discuss their breakthroughs and highlight multidisciplinary approaches to key areas of research. We do this to help bring forth the discoveries needed to make a healthier, better world. Our partnership with you, the scientific community, helps us all to reach the incredible. So with that, let me jump into the presentation. Today's presentation is broken up into 4 parts. I'll start out by giving some background about the current status of neurobiology research and what is needed for neurological models. I'll then give an overview of ATCC neural progenitor cells, including quality control data, biological markers, and how we monitor the differentiation of these cells. I'll then show you how these cells can be used in toxicology studies, either in their undifferentiated state or after differentiation in ATCC dopaminergic neuron medium. First, a bit about the challenges that neuroscientists are facing, especially when studying Parkinson's disease. In the current state of in vitro neurobiology research, models come from either primary cells from animals, such as mice and rats, continuous cell lines that are usually isolated from tumors, and iPSCs, either commercially acquired or self-made. Now, there are problems with building your models of neurobiology from all these sources. Primary animal neurons have wide donor variation. In addition, no whole animal model completely recapitulates the clinical symptoms and pathology of Parkinson's disease that's seen in humans. And that suggests that rodent-derived cells would also not fully capture the human disease state in vitro. Continuous cell lines are generally not isolated from normal cells and thus may not be predictive of the in vivo situation. These cells may have lost their in vivo biologies, reducing their applicability to your study. They may have been altered genetically as well to immortalize them, which further reduces their physiological relevance. And creating iPSCs and then neurons from iPSCs is very time and labor intensive. It can take from 8 to 10 weeks to to create the intermediate neural progenitor stage. Then there is the additional time that you need to take to differentiate them. There's also the problem of securing iPSCs from Parkinson's disease patient tissue. So what is needed for in vitro neurological studies are biologically relevant models of normal and diseased tissue, which have been validated for neural functioning and are predictive for screening applications in drug development, normal physiology research, and toxicological studies. Neural progenitor cells answer all of these needs, as I'll show you in the following slides. I've shown this slide in previous webinars, but to help us understand how neural progenitor cells can be the ideal model for neurotoxicology and other studies, I'd like to take you through the process of neuronal differentiation. By the way, sometimes I'll be switching back and forth between neural progenitor cells and the abbreviation NPC. Now, in vivo, neural stem cells undergo asymmetric cell division cycles, which give rise to either more neural stem cells or to neural progenitor cells. The neural progenitor cells may then differentiate down different lineages that have specialized morphologies and functions and develop the marker expression of those neural lineages. Neuronal differentiation from iPSCs is also characterized by these changes. At ATCC, we've established a process that can generate an unlimited supply of neural progenitor cells from normal and disease-induced pluripotent stem cell lines via the formation of embryoid bodies. iPSCs express TRA160, which we use as a marker of their pluripotent state. Over the course of 4 to 8 weeks, when cultured in our proprietary NPC expansion medium, the iPSCs form three-dimensional aggregates called embryoid bodies. These are broken up and the cells are replated in the neural progenitor cell expansion medium. Subsequently, the replated cells differentiate into neural progenitor cells. During this differentiation process, TRA160 expression is lost and replaced by nestin and PAX6, which are widely regarded as markers of neural progenitor cells. Now, expression changes again when neural progenitor cells differentiate further into their lineages. And that can be achieved by culturing the cells in lineage-specific medium formulations. Neural progenitor cells that differentiate into astrocytes lose expression of nestin and PAX6 and instead produce glial fibrillary acidic protein, also known as GFAP. Neural progenitor cells that differentiate down neuronal sulfate express microtubule-associated protein, or MAP2, as well as double cortin, which is abbreviated as DCX. They also express neuron-specific class 3 beta-tubulin, or 2J1. Neuronal cells can further differentiate into dopaminergic neurons and are identifiable by expression of tyrosine hydroxylase, or TH, which is the enzyme that's involved in dopamine formation. Finally, neural progenitor cells that differentiate into oligodendrocytes express the marker O4. So implementing neural progenitor cells into your experiments can save 1 to 2 months worth of work and gets you running your experiments on differentiated neurological cells sooner. Furthermore, these neural cells will faithfully recapitulate the in vivo situation. So how are neural progenitor cells a better model? Well, first of all, since they're derived from human-induced pluripotent stem cells, NPCs are human models. They exhibit no donor variation because they're derived from a single donor iPSC. Since many of our NPC offerings contain reporter constructs, live imaging is possible. NPCs are flexible in that they exhibit full differentiation spectrum. In other words, they can differentiate into neurons, astrocytes, or oligodendrocytes. In addition, to simplify things for the culturist, ATCC has a complete system of cells supported by media kits that enable the expansion and differentiation of its neuroprogenitor cells. ATCC also provides protocols to help you with your expansion and differentiation processes. So, how will you see more meaningful results in your research by including NPCs? They give more biologically relevant results and provide a more predictive system because they're derived from the same parental cell. Parkinson's disease neuroprogenitor cells provide a model that could better replicate the disease state, but in vivo. Also, neuroprogenitor cells express lineage markers for easy endpoint readouts. In addition, using the reporter labeled cells ensures lineage identity. As mentioned above, neuroprogenitor cells can differentiate down neuronal or glial pathways, allowing you the flexibility of studying more than one subtype of the nervous system. Our NPCs are easy to use because we provide a complete system of expansion and differentiation media, as well as the well-tested and validated protocols. Because you can expand your neuroprogenitor cells, there's the potential to have more replicates in your experiments, allowing you to be much more confident in your results. And finally, and probably most importantly, utilizing neuroprogenitor cells saves time over creating neural cells from iPSCs. You save about 8 weeks of embryoid body formation. You can work with your differentiating or terminally differentiated neuroprogenitor cells sooner, and your experiments will yield results faster, allowing you to evaluate your hypotheses faster. ATCC has several neuroprogenitor cell offerings, including neuroprogenitor cells derived from normal bone marrow CD34 positive iPSCs. These include ACS-5003, which is one of the subjects of our study today. I'll also be focusing on ACS-5001, which are neuroprogenitor cells that were derived from iPSCs created from a patient with Parkinson's disease. We have reporter cell lines, which can be used to monitor late neuron differentiation, astrocyte differentiation, and early neuron differentiation. One of these has GFP marker linked to double cortin expression. Another has a nanoluck HaloTag reporting GFAP expression. And ACS-5007 has a MAP2 nanoluck HaloTag reporter, which informs early neuron differentiation. During this presentation, I'll also be focusing on this cell line. We also carry a growth and expansion kit and dopaminergic neuron differentiation kit. To use these, you simply add the contents of the kits to DMMF12 to make your complete medium. I should also point out, as part of our effort to provide better biological model, we perform full characterization of ATCC neuroprogenitor cells. We do tests such as post-thaw cell count, cell longevity, neuroprogenitor cell marker expression, and neuronal differentiation potential, which I'll be focusing on shortly. We also perform standard cell biology QC testing such as STR profiling, sterility, mycoplasma, and a viral panel. So I've given you some background information about iPSC-derived neuroprogenitor cells. I'd like to guide you through the differentiation of these cells next. I'll show you a lot of marker expression data to convince you that neuroprogenitor cells can be used to model phenotypical astrocytes, oligodendrocytes, neurons, and specifically dopaminergic neurons. We first took a look at the expression of the positive markers of neuroprogenitor cells, nestin and PAX6, and the negative marker TRI160. These neuroprogenitor cells were incubated in ATCC expansion media, as mentioned earlier. And as you can see, the ATCC neuroprogenitor cells express nestin, and PAX6, and lacked expression of TRI160. So according to our criteria, we have phenotypic neuroprogenitor cells. So next we'll look at the expression of those markers in Parkinson's neuroprogenitor cells. These cells were incubated in ATCC expansion medium, as in the cells in the previous slide. As you can see, the Parkinson's disease neuroprogenitor cells express nestin in green and PAC6 in red, and lacked expression of TRA160. By the way, the Parkinson's neuroprogenitor cells also express SOX1, a marker of activated neural stem cells. So the Parkinson's neuroprogenitor cells also fulfilled our criteria for phenotypic neuroprogenitor cells. We then wanted to confirm that our dopaminergic neuron media could induce differentiation of neuroprogenitor cells. Normal human NPCs, here ACS-5003, the non-reporter line, were seeded on cell matrix basement membrane gel-coated plates and cultured in NPC expansion media overnight. We then incubated the neuroprogenitor cells with dopaminergic differentiation media for up to 3 weeks. This, by the way, is the treatment parameters for the other dopaminergic differentiation studies I'll show you for this particular cell line today. The immunostaining of tyrosine hydroxylase that you can see in the right panel and 2J1 in the left panel indicate that ATCC neuroprogenitor cells had the potential to be differentiated into 2J1 positive early neurons and tyrosine hydroxylase dopaminergic neurons. We then wanted to confirm that our dopaminergic neuron media could induce differentiation of Parkinson's disease neuroprogenitor cells. Here, the ACS-5001 cells were seeded on cell matrix basement membrane gel-coated plates like the normal NPCs and cultured as before in the dopaminergic neuron media for 3 weeks. By the way, slightly different from the normal NPC differentiation protocol, the differentiation media for these cells contained 5 micromolar CAR99021, which is a potent GSK3 beta inhibitor known to strongly activate Wnt signaling. It turns out that this component was required for the differentiation of the Parkinson's NPCs. So you can see that immunostaining here indicates that the Parkinson's neuroprogenitor cells had the potential to be differentiated into 2J1 early neurons, here on the left panel, and on the middle and right, Th1 positive dopaminergic neurons. Now, since neuroprogenitor cells can reportedly differentiate into glial and oligodendrocyte cells, we looked at the expression of markers of those cell fates. In this study, we compared normal iPSC-derived and Parkinson's disease fibroblast iPSC-derived neuroprogenitor cells. We incubated groups of each of these cells with in-house oligodendrocyte differentiation media for about 8 weeks, and other groups of neuroprogenitor cells with commercially available Astrocyte differentiation media for about 5 weeks. If we look at the upper panels, what we see is similar staining and morphology for the astrocytes of both cell types. Now take a look at the lower panels. While the staining was similar for oligodendrocyte lineage differentiation, the morphology in the Parkinson's disease-derived NPCs was a bit different. So our normal neuroprogenitor cells were definitely able to show phenotypic markers of astrocytes and oligodendrocytes. The Parkinson's NPCs astrocytes look similar to the normal neuroprogenitor cells. The difference in the Parkinson's and the normal oligodendrocytes could be due to the disease origin of the Parkinson's disease NPCs. In fact, lately it's been hypothesized that oligodendrocytes are somehow involved in the Parkinson's, the pathogenesis of Parkinson's disease. Now, as an aside, I'll show you the complete range of what we have for neuroprogenitor cells. Here we looked at the dopaminergic neuron differentiation of the NPC reporter lines. Immunostaining of tyrosine hydroxylase and 2J1 indicate that all 3 neuroprogenitor reporter lines had a potential to be differentiated into early neurons, where we see 2J1 stained in green, and tyrosine hydroxylase dopaminergic neurons, which are stained in red, after a 3-week incubation with ATCC dopaminergic differentiation media. So all 3 reporter lines can be differentiated into dopaminergic neurons. In addition to regular NPC features, we can quantitatively monitor the process of lineage-specific differentiation of the 2 nanologue HaloTag reporter lines. As shown in the top panel, nanoluciferase secretion in dopaminergic culture media of the MAP2 reporter NPCs significantly increased in a time-dependent manner. So treating the MAP2 reporter NPCs with ATCC dopaminergic media resulted in a marked increase in luciferase secretion, and it was observed at 4 days and sustained up to 18 days. Likewise, in the bottom panel, we can see nanoluciferase secretion in GFAP reporter NPCs in a time-dependent manner. There was significant increase actually in luciferase at day 16. Therefore, these neural progenitor cell reporter lines can be used to assess the progress of NPC neural differentiation. In addition to monitoring GFAP or MAP2 expression by measuring luciferase activities, we can quantitatively track GFAP or MAP2 expression by using immunocytochemistry with HaloTag antibodies, or by live imaging with fluorescence-labeled HaloTags. Here in the bottom panel, we show expression of HaloTag and MAP2. In the middle panels, we show expression of GFAP and HaloTag. In addition, the HaloTag expression was co-localized well with GFAP or MAP2. Now, the top panel shows the time-dependent expression of GFP in the double-quartin PGFP reporter line after incubation in the dopaminergic media. Therefore, we can monitor the process of mature neuron differentiation by live cell imaging. So these neuroprogenitor cell reporter lines provide a powerful tool for studying lineage-specific differentiation and for drug screening. So we looked at the differentiation potential of ATCC neural progenitor cells via immunocytochemistry and reporter expression. We wanted to confirm those data by looking at the gene expression of neural progenitor cells after differentiation in our dopaminergic neuron media. In addition, in the natural in vivo environment, many different types of neurons can be formed from the same growth factors in our media. Therefore, we couldn't ignore the idea that other mature neural types could be formed or found among our differentiated dopaminergic neurons. Aside from dopaminergic neurons, we tested for the expression of markers of glutamatergic, GABAergic, motor, and cholinergic neurons. We chose the PD-NPC line ACS-5001, the normal neural progenitor cell line ACS-5003, and the MAP2 reporter line ACS-5007 to look at the gene expression of these markers after dopaminergic differentiation. For these experiments, all 3 cell lines were treated with the dopaminergic differentiation media for 0, 1, 2, or 3 weeks. RNA was then extracted and cDNA was synthesized for qRT-PCR analysis of neuronal gene expression. And we used TACMAN probes for the genes that are shown here. So first we confirmed the upregulation of early and dopaminergic neurons associated RNA sequences. Here we see that messenger RNA coding for the functional neuronal marker MAP2 increased significantly during 3 weeks of neuronal differentiation. In the left panel, ACS-5003 and ACS-5007 NPC lines. In the right panel, we also saw this in the ACS-5001 Parkinson's disease cells. So this confirmed our immunocytochemistry and reporter activity evidence that I showed you previously. Similarly, 2J1 messenger RNA increased significantly during 3 weeks of neuronal differentiation of all 3 cell lines. Again, the normal and reporter lines are in the left panel, and the Parkinson's disease neuroprogenitor cells on the right. So here's further evidence that the progenitor cells are differentiating into neurons. In addition, from 2 to 3 weeks, there was a significant increase in tyrosine hydroxylase messenger RNA in all 3 NPC lines. Remember, tyrosine hydroxylase is the enzyme that converts L-tyrosine to L-DOPA, which is critical for dopamine production. We also see increases in the developmental transcription factor NURR1 mRNA expression. Because there is little increase in the expression of these genes in week 1 and week 2, this shows that neuroprogenitor cells must differentiate for at least 3 weeks before we see mature midbrain dopaminergic neurons. The expression seems to be lower in the Parkinson's disease cells, but it's still significant. Related genes like VMAT and DAT with opposite functions also increase significantly. For example, vesicular monoamine transporter 2, which we've abbreviated here as VMAT2, is a membrane protein that pumps dopamine and other transmitters from the cytosol to synaptic vesicles. We saw increases over the course of 3 weeks, and this is in all 3 neuroprogenitor cells. Also, dopamine active transporter, or DAT, which pumps dopamine out of the synapse back into the cytosol, we saw significant increases in. These increases, however, did seem to drop off after the first week. Finally, we looked at expression of aromatic L-amino acid decarboxylase, or ADAC, the enzyme that converts L-DOPA into dopamine. We saw that this enzyme also significantly increased in a time-dependent manner in the normal NPCs on the left and the Parkinson's disease NPCs on the right. So I've just showed you the expression of 3 neuron cell fate genes and 4 dopaminergic neuron genes increased in neuroprogenitor cells after 3 weeks incubation in the ATCC dopaminergic differentiation media. So this, the immunocytochemical data and the reporter expression data indicate that we reliably get dopaminergic neurons from this media. We now want to test what other types of neurons this media could stimulate the neuroprogenitor cells to differentiate into. So this table essentially summarizes what all we saw. First, I'll focus on the glutamatergic gene expression. Glutaminase 2, or GLS2, catalyzes the formation of glutamate from glutamine. It and the vesicular glutamate transporters VGlut1 and VGlut2 are markers of glutamatergic neurons. We saw that the messenger RNA of 3 of these markers was increased in all 3 cell types. We then looked at the expression of a receptor of gamma-aminobutyric acid, which is also known as GABA. We saw that messenger RNA increased significantly in normal and Parkinson's NPCs in a time-dependent manner during neuronal differentiation. So that suggests that we have GABAergic neurons in our cultures as well. We also looked for upregulation of motor neuron genes such as LIM3, HB9, and N1. We also looked at the cholinergic neuron gene CHAT. There was significant increase in LIM3 in the normal and Parkinson's disease, but not in any of the reporter line. Like LIM3, HB9 messenger RNA was increased in the ACS-5003 and but was unchanged in the ACS-5007. It was elevated also in ACS-5001, the Parkinson's line, but the expression dropped off after the initial spike. N1, the expression rose after 3 weeks in the normal line. In the Parkinson's line, expression rose after 1 week, but gradually dropped off. And no significant expression was seen in the reporter line. Chat expression in the reporter line rose after 2 weeks. In the normal line, the expression never seemed to get off the ground. And in the Parkinson's disease line, expression rose after 1 week and rapidly dropped off. So generally speaking, the neural progenitor cells showed an increase in the messenger RNA of a range of specific neuronal markers after incubation in the dopaminergic differentiation media. We saw the most consistent increases in dopaminergic neural markers, which signified the presence of those neurons. We also saw differing levels of other neuronal markers, suggesting that we have a heterogeneous population of neurons within the cultures. So in the next slide, I'll show some immunocytochemistry data that confirms the presence of other types of neurons during the dopaminergic differentiation. We did immunocytochemistry using tyrosine hydroxylase, NURR1, GLS2, VGLUT2, CHAT, and 2J1 antibodies in ACS-5007 neuroprogenitor cells. These were differentiated into neuronal cells using the dopaminergic media for 3 weeks. So in this slide, I'm showing that consistent with the QRT-PCR data, we saw staining of all of these neuronal markers. In the top row, we see NURR1 co-localized with tyrosine hydroxylase. In the middle row, we can see CHAT, which is also co-localized with 2J1. And in the bottom row, there's GLS2 and VGLUT2 staining. So these experiments support the messenger RNA expression studies that I showed you. So I've showed lots of data supporting the directed differentiation of these cells. Now I'd like to show you the value of these cells for toxicological studies. So this first battery of experiments I'm going to show was in undifferentiated normal neural progenitor cells. And these cells were used at passage 6. The cells were treated with compounds for 2 days after plating an ATCC neural progenitor cell expansion medium. They were treated for 2 days with 1, 10, and 100 micromolar concentrations of each drug. And then they were incubated with a rizazurin-based viability assay for 6 hours. This figure shows the effect of several potential neurotoxins on the ACS-5003 neural progenitor cells. So we treated the cells with amiodarone, which is associated with peripheral neuropathies, chlorhexidine, an agent recently identified to be neurotoxic to neurons, SHSY5Y cells, and Schwann cells, and the acute and chronic toxicant digoxin. Also, in this rogues gallery, we have paclitaxel, a microtubule stabilizer known to cause neuropathies, cisplatin, a platinum-based apoptotic agent known to have neuropathic effects, but not reputed to be neurotoxic, and vincristine, a plant alkaloid shown to cause peripheral neuropathy when used to treat pediatric ALL patients. Piperine is a nociceptive agent, which is not known to cause neurotoxicity. In this experiment, each of the compounds, except for piperine, displayed toxic effects on the neural progenitor cells. However, cisplatin and hydroxyurea was toxic only at the maximal dose of 100 micromolar. Paclitaxel and vincristine appeared to have the most toxic effects, which was absorbed at the lowest concentration of 1 micromolar. We also saw some different results in the undifferentiated Parkinson's disease neuroprogenitor cells. So the ACS-5001 cells were resistant to cisplatin, piperine, and to all concentrations of hydroxyurea. They were sensitive at all concentrations to paclitaxel and vincristine. They also displayed sensitivity to amiodarone and chlorhexidine. To confirm what we saw with paclitaxel, we did a dose response curve using over 7 different concentrations of the compound. These experiments were done in both of the non-disease neuroprogenitor cell lines. The neurotoxic effect of paclitaxel was similar in both of the lines. So the IC50 of paclitaxel on ACS-5003 was 0.9 micromolar, and in ACS-5007 it was 1.6 micromolar. So I just showed you data using undifferentiated neuroprogenitor cells. We also did neurotoxicity studies with the neuroprogenitor cell-derived neurons. In this set of experiments, we used reporter-labeled ACS-5007 and differentiated them in dopaminergic media as before. Unlike the undifferentiated neuroprogenitor cells, the differentiated neurons were resistant to paclitaxel. Now, the difference in sensitivities could be due to the fact that paclitaxel may be toxic only in dividing cells. Nevertheless, neuroprogenitor cell-derived neurons were also sensitive to vincristine, amiodarone, and chlorhexidine. We confirmed what we saw in the differentiated neurons in this study. We used high-content imaging analysis to look at the neurotoxicity of 10 micromolar paclitaxel, cisplatin, and chlorhexidine in the normal neurons in both the ACS-5003 and ACS-5007 derived neurons. We treated them for 2 days and then stained with calcium green AM and HUXT. On the left, we show viability. On the right, NURRI outgrowth. So consistent with the viability data, 10 micromolar of chlorhexidine significantly inhibited the NURRI outgrowth, but we saw no effect with paclitaxel and cisplatin. And it appeared that also we saw a reduction in viability with chlorhexidine, but also, again, no change in viability after paclitaxel and cisplatin treatment. So I put this table together to summarize our toxicology study. On the first column of the table, we show a list of the toxins that we tested. ACS-5003 neuroprogenitor cells were sensitive to most of the compounds that they were treated with the exception of piperine, which we actually expected that, as piperine's only a noise-susceptive agent. Now, cisplatin and hydroxyurea caused only mild toxic effects in the Parkinson's disease neuroprogenitor cells. The amiodarone, chlorhexidine, vincristine, and paclitaxel did exert toxic effects in the Parkinson's neuroprogenitor cells. The Parkinson's disease neuroprogenitor cells were resistant to piperine, as well as the compounds that the normal NPCs were only mildly resistant to, or mildly sensitive to, rather. Unlike the neuroprogenitor cells, the ACS-5007 and ACS-5003 derived neurons were resistant to paclitaxel. Nevertheless, the neuroprogenitor cell derived neurons were also sensitive to vincristine, amiodarone, and chlorhexidine. So I've introduced neuroprogenitor cells. I've gone through their differentiation potential. I've shown how undifferentiated neuroprogenitor cells and differentiated neurons can be employed in toxicological studies. Now I'm going to summarize the talk. ATCC has created neuroprogenitor cells. We developed both normal non-reporter and lineage reporter lines, as well as a Parkinson's disease iPSC-derived line. We also offer media supplements for expanding your neural progenitor cells and differentiating them into dopaminergic neurons. Our neural progenitor cells are a human model of the nervous system, and because they're derived from iPSCs, there's no donor variation. As I mentioned, you can expand and bank these cells. The cells can differentiate across a wide spectrum of neural and glial lineages. We have dependably differentiated our neural progenitor cells into neurons, astrocytes, and oligodendrocytes. Live imaging of differentiation is possible with our GFP expressing reporter line that signals endpoint neural differentiation. Our studies demonstrated that after treatment with our dopaminergic differentiation media, normal and Parkinson's disease neural progenitor cells have the potential to be differentiated into dopaminergic neurons, GABAergic neurons, glutamatergic neurons, motor neurons, and cholinergic neurons. And finally, I showed you that ATCC neural progenitor cells are suitable for drug toxicology screening applications using a panel of 7 drugs with varying levels of neurotoxicity. So one last word before we move into the question and answer session. Let's continue to cultivate this collaboration. You can help us elevate our better biological models, which will enable greater specificity and functionality in your experiments. Join our community of early adopters. To learn more about becoming an early adopter of neural progenitor cells or one of our other advanced biological models, visit 2020.atcc.org. Our partnership with you, the scientific community, allows all of us to reach the incredible. And now I will take your questions.
Thank you, Dr. Shapiro. In just a few moments, we will begin our question and answer session. Please use the chat function available through the webinar program to submit questions. This session will be documented as a web page and archived along with a recorded webinar presentation on the ATCC website at www.atcc.org. So for our first question, often neuronal cultures require the use of plates coated with a substrate such as poly-D-ornithine or poly-L-lysine. Do NPC cultures require a gel coating or substrate?
Thanks, Kara. Actually, that's a very good question. They do. The neural progenitor cell protocols require that the culture dishes be coated with cell matrix membrane, basement membrane gel, which is ATCC part number ACS-5003.
Our next question is, do you have disease NPC lines?
So at present, the only neural progenitor cell disease line that we have is the Parkinson's disease iPSC-derived line.
Another question is, how many passages can these cells undergo?
So our neural progenitor cells can be proliferated at least 15 PDLs, and that's population doubling levels, and passaged over 20 times. Some of the results that I showed today were using cells from passage 10, actually.
Another question is, can you explain why you are getting different neuronal populations when you claim your media is dopaminergic differentiation media?
It's actually a really good question. And the answer is the majority of the growth factors in our differentiation media are common to generate all neuronal cell types. The big difference is the concentration that's needed to make a majority of a specific neuron type in a specific culture dish. So by tweaking the concentration of growth factors, we're able to increase the number of dopaminergic neurons relative to other neuron types.
Another question is, what dissociation reagent is recommended for the passaging of NPCs?
So ACS-5001 neural progenitor cells require undiluted ACCutase for passaging. And that's actually different from the normal neural progenitor cells, which use ACCutase diluted 1-to-1 with PBS.
So you mentioned the addition of CHIR 99021 to the media for differentiating the Parkinson's disease-derived NPCs into dopaminergic neurons. Where did you get the compound from, and what concentration is it used?
Okay, so for differentiating ACS-5001 neural progenitor cells to dopaminergic neurons, you have to add the CHIR 99021 to a final concentration of 5 micromolar. We get the compound from StemGent. It's cat number 04-0004-10. And the component can be added in an aliquot of dopaminergic neuron media and stored for 4 degrees C for about a week.
Our next question is, what is the optimal cell confluence when passaging NPCs?
It's a good question. Unlike other primary type cells, passaging neural progenitor cells near 100 confluence does not affect cell performance. Actually, we highly recommend to passage NPCs when they reach about 95% confluence. Do not split the neural progenitor cells when they're less than 85% confluent. That's actually will inhibit the cell's proliferation.
So it looks like we have time for one last question, and that is, are there passaging recommendations for NPCs during tri-lineage differentiation?
Okay, well, it's okay to split cells during astrocyte and oligodendrocyte differentiation when the neural progenitor cells become confluent. However, passage neural progenitor cells will not survive well during dopaminergic neuron differentiation. Overconfluence of the neural progenitor cells will not affect the outcome of dopaminergic neuron differentiation.
At this time, we will conclude our question and answer session. I would like to thank Dr. Shapiro for the excellent presentation, and thank you everyone for attending the webinar. Any questions that were not answered this afternoon will be answered and posted with the video at www.atcc.org. Thank you again and have a great day.